Okay, so hi everyone. Today's lecture is going to be about protein structure classification and evolution. So the basic principle is that we want to be able to compare structure and to classify them to know more about their evolutionary history and their function. So if you have the same structure, you expect to have similar function. So the very basic principle for a protein to fold in solution is the hydrophobic force. Because side chains are hydrophobic, they will tend to form a hydrophobic core, which is protected from the solvents. The problem is, if you do this, you will also have to fold the main chain atoms. And these are they're polar, so they're hydrophilic in general. So the solution found by proteins to do this is to form secondary structures. The secondary structures are stabilized by hydrogen bonds between polar atoms, and because of this stability, the protein is able to form these structures, these 2D structures that can arrange into a core, which will be, so that's just a way, let's say, the hydrophilic atoms interact with water, and they will exchange this interaction for hydrogen bonds within the protein, and this will be energetically favorable to do this. And that's how the packing is going to be formed. So very important, one of the main secondary structure elements is the alpha helix, and one thing that is interesting about this one is that it was proposed before anybody had seen alpha helix in real. So it was really the model before the observation here. So we have 3.6 residues per turn, meaning that residue I interact with residue I plus 4. The second very main secondary structure element is the beta sheet, which is formed by several beta strands. It's not really flat, it's kind of pleated, right? And you have two types, either parallel, where all the beta strands are in the same orientation, or anti-parallel, where the beta strands alternate their position. There are also tons of loop structures, so if you think of a loop, you may say it's very flexible, but it still has a structure, so it's not random coil, it's structured, it's constrained. So if you have a 3D structure of protein, you may assign the secondary structure by defining some geometrical properties, like you want some distance, you want some angles. So for example, the DSSP program does this. It has a vocabulary, a dictionary of secondary structure elements, and it will just assign the 3D coordinates. Based on the 3D coordinates, assign every atom, every residue to a secondary structure type. This is very different from predicting the secondary structure starting from the protein sequence. So there is a secondary structure prediction and just assignment based on the 3D coordinates. So here you have an example of two proteins where you see the 3D structures are very similar, and on the right you have the topology diagrams which represent the connectivity of the secondary structure elements. And here you see that you have an opening of the chain which is in very different position. The C and N show you the N-terminal and C-terminal extremities, and they are located in different places. So this means that, in fact, the connectivity of the secondary structure elements is not the same. So these proteins may be classified in different folds, and it's not obvious from looking at the 3D structures that they are different. So something a bit bigger than the secondary structure element, basic combinations that you can do with them define structural motifs. So for example, in the practical, we looked at coumadline, and this protein has a very known motif which is called an EF hand. And this is the helix motif, which binds to a calcium ion. So when it's not bound, it's like this, and when the calcium binds, then it opens. If you go a bit bigger, you will find the notion of protein domains. So a domain is a fundamental unit of tertiary structure. So here you have an example of domain compositions for several receptor tyrosine kinases. You have essentially an extracellular part, which can vary a lot in domain composition, an intracellular part, which is always the kinase itself, so the enzyme, that will always perform the same function, catalyze the phosphorylation of some tyrosine residues. So by combining these different domains, you can have very different proteins doing essentially different things in the cell. So the notion of domain is a bit ambiguous and poorly defined, like all the basic notions in biology, if we can say that. So should we consider it's a structurally distinct region, a compact unit? We can also think of it in terms of evolution. Is it an evolutionary unit? Is it something that can fold, so that can just adopt the 3D structure irrespective of the rest, independently for the rest? Or is it something that has a particular biochemical function, let's say, for example, an enzyme? So in the past, proteins domain have been described in terms of all these elements: structure, compactness, function, evolution, folding. But it's not very clear what we should choose. So depending on the context, I would say a domain may be one or the other thing. And different databases will give you different references. So usually when you have a small protein, you will consider it's just single domain protein. And one thing that we can say is that in any case, a domain will be formed by a combination of secondary structure elements, so a special connectivity of these elements and motifs. So there is basically a jungle of shapes, and the classification of protein structures is based on these assemblies, combinations of secondary structure elements. So the most basic level is just the composition in secondary structure elements. So you can be all alpha, all beta, a mix of the two. So either a mix of the two separated or a mix of the two alternating, okay? So here I will let you go through the slides on your own because I think these are interesting cases, but I don't want to spend too much time. It's just to give you an idea of all the folds. So a fold is a combination of secondary structural elements, all the folds that are known, that are ancient, how they were found. This is something more for some general, and also what are their differences. Let's put some order now. So this I will talk a bit about the taxonomy. So there are several ways of classifying protein structures. So the most basic class level, as I said, is this secondary structure content simply. And then it's a hierarchical taxonomy. So then for a given class, you will have several folds or topology, which represent the global shape of the protein. And within each fold or topology, you will have several superfamilies. And now when we are at the level of superfamily, we think of evolution. So proteins that are structures that are in the same superfamily have some evidence that they're related in evolution. So there is evidence that they come from a common ancestor. So here, simply an example, you have a O beta on top and then alpha beta at the bottom. So this is the class level. So then for the fold, major secondary structure in the same arrangement, that's the idea, superfamilies, evolutionary notion. So for example, here you have an example of PA superfamily, where we highlighted the beta barrels, beta sheets in red. So you see that there is some similarity between the two, but of course many things still vary. So there is a lot of variability within a superfamily. So the resources that are available for this are mainly Cath and Scope. So Cath is mainly automated and Scope is mainly manual. So there has been a lot of effort, I don't know, maybe 20 years ago, 30, 20 years ago, to construct these databases. And many people were annotating this manually, especially for Scope. And around 2009, 2010, let's say, there's been a big decrease in the investment in these classification. So you will read sometimes that there is no new fold that is discovered, that everything is known about the folds in these databases. But I think that there is a huge human bias here. It's just because we stopped investing, I think, in these things. And now it's more automated, and it's the new structures tend to populate the same folds that we know. So the numbers are here in the different databases. You must keep in mind that there are inconsistencies between the databases. They do not fully agree. And so it's Also human biased, even if it's automated, so it's not very clear what should be taken as really the reference. The ECHO database is on the bottom is much more recent. So here it's the focus is given on remote homology, and it comprises the biggest number of PDB entries, I think. Okay, so standard truths, just to compare the topology between scope and CATH, the classification. So in scope you have fold, in CATH you have architecture, which is not fold. It's between, so I would say the topology of CATH is kind of similar to the fold scope, and the architecture is something additional. And but you see superfamily, homologous superfamily, it's like similar, but the definitions are slightly different. And E-code extends scope by adding remote homology relationships. So that was what I was talking about. So there is some notion of architecture here and some remote homology ensembles. So it's okay, hierarchy. So you can see here an example. We take a class, alpha and beta, and then we show 3 folds. Well, not fold because it's CATH. So 3 architectures. And then we take the sandwich architecture, we have 2 topologies, so you can see the level of difference we have. So scope E was an effort to revisit scope and make it more automated, also more complete. So there are new clouds that are found, so still we can find new clouds, new fold, new things. This is an example. And there is also some effort to develop scope 2, which will be non-hierarchical. So just transform this hierarchy by a network with much complex relationships. So it's kind of an ontology, if you wish. And they want to increase the annotation on the ontology. Something like this with a relationship. But I think it's ongoing. It's really not finished. So that's the idea. So then we can talk a bit about the protein structure space. So there's been a lot of efforts in years 2005-6 to really try to see how protein structure populate the space. So here it was a PNA study in 2006 where you compute pairwise distances and then multidimensional scaling and project onto the 3 first modes. So the idea is that the result of this study showed that the protein structure space is very sparsely populated. So that's a bit like sequence. We observed that among all that is possible in terms of 3D conformation of geometry, there is a few parts of this space that is really populated by proteins, and it's the same. You can have many sequences. The combinatorics is huge. Even if we have access now to a huge amount of sequences, we realize that the space of sequences that is really populated is small compared to the whole space. So now let's go back through time. Present-day protein space is the result of 3.7 billion years of evolution constrained by the underlying physico-chemical qualities of the protein. So there are 3 things that you should keep in mind. The evolution of proteins is not the same as the evolution of organisms. Also, structures tend to diverge less than sequences. So if you have 2 sequences that share 40% of sequence similarity, of identity, they will have the same structure basically. And there exists no remain of primitive proteins though, so we are always, trying to infer, trying to predict what was there before. But what we see now, even ancient folds of ancient origins, they have evolved, and what we see is just present-day proteins, and this should be clearly kept in mind. Okay, so let's take an example, an analogy with fishes. So let's say we have these 2 fishes at the bottom, which may arise from a common ancestor. But maybe with this level of similarity between them, they arise from 2 different ancestors. So that's a big question with the protein structures. Do they come from the same ancestor, from a common ancestor, and in that case, we're in the presence of divergent evolution, or do they come from different ancestors? And this is convergent evolution, meaning they really look alike, but the evolutionary paths are completely different. So evolutionary processes are really distinguished between these two: divergent evolution, convergent evolution. We will talk about homology in one case, analogy on the other case. So there are ambiguous cases like this one where we don't know exactly. You have something that is similar, two strands and a helix, but the arrangement between them is different. So is this analogy? Is this homology? These things are not really decided. So the basic principle for homology is that we have transitivity. So if A and B are homologous, meaning they descend from a common ancestor, then if B and C are homologous too, then A and C are homologous. So you have a transfer of homology. So it means that you can go far in sequence identity and still have some homology insight or evidence because of this transitivity between intermediates that are closer. So in 2010, so around these years, 2000, there was also this notion developed of the galaxy of folds. So here you have a representation of clusterization of the folds. And what was shown here was that many tight clusters were composed of several superfamilies of the same folds, suggesting a common origin. So at first I would say researchers were thinking that a lot of convergent evolution was at play and that we had common ancestry really at the level of superfamilies, but then by comparing these folds it became more and more obvious that in fact you could have a common origin for many of these of the folds at higher level than superfamilies. So it's still, these things are still open questions. We don't really know. But I would say that in recent years, what has become obvious is that you can have remote homology, meaning things that look really far away from each other, but still they descend from a common ancestor. So here it's an example where you see evidence of homology between folds. So these are two different folds. So first you see they're not so different, right? But still in the classification, they're really distinguished. So what's the difference? Well, you have the SAM MTAs on the left, which can be seen as a Rossmann fold, Rossmann domain, what you have on the right, with an insertion of a strand. So the strand is in orange, is an invasion, right? And you also have the helix in the Rossmann fold in purple, which is which disappears in the SAM. Okay. So here, when you see this, you may think, okay, where does this come from? It's not so different, so maybe there's a common ancestry, and just at some point this strand was inserted. Here is another example of the K homology module that you have on top. So you can have this homology module which is in mini form, maxi form, or not at all. And so here the question again is, this is a widespread RNA binding motif, but in principle these also proteins represent different folds. So here it's not clear again whether we have some remote homology, or whether they descend from a common ancestor, or whether it is convergent evolution. So you see the difference is that you can see the difference with the colors and also in the sequence. You have some sequentiality on top between the parts in green, whereas the sequentiality at the bottom is between the parts in blue, right? So in green on top you have the central strand that is green, and at the bottom it's the left strand that is blue. So you can see also in the sequence the green is at the C terminus while the blue is at the N terminus. So you have this different positioning with respect to the sequence, but when you look at them, they really look the same, right? Except for the no KH, which are really different, and they miss the helix. So here also there was series proposing to go from one to the other, whether this is homology or not. So that's a big question. Can protein structure evolve? Can we go from A, what you see in A, to H? So this is a scenario that is proposed. Okay, let's say I have many sheets. I have all just beta sheets. I start to remove some, I start to transform some into helices, and then I end up with something fully helical. Is it possible, going from fully beta to fully alpha? So here along the way, this is speculative of course, but along the way you have real PDB structures, right? These proteins are real. And it's kind of shows a plausible scenario. So how do we compare structures to be able to classify them? The most popular measure is the root mean square deviation. We talked about this in practical. So basically let the average deviation between the two structures, and it's average over an ensemble of atoms that are matching between the two structures. So you need a correspondence, an exact correspondence. Another score, which is another metric, which is used alternatively as an alternative to the RMSD, and sometimes it performs better. It depends again on the context. At least it is less sensitive to local structural variations, is the TM score. So here the magnitude of the TM score for a random pair is length independent because we have a normalization factor. So it was defined in 2004, and so if you have a score below 0.2, you have randomly chosen unrelated protein structures. A score better than 0.5 assumes the same fold. Okay, so because of this normalization by the length, you are less sensitive to structural variation. So these values that you see were determined imperfectly. So this TM score is used typically to also estimate that the quality of models of structures compared to experimental gold standard structures. And then you have tons of metrics that are defined especially by these people who are interested in predicting protein structures. So you have the CASP experiment, which is an international challenge where teams from all around the world try to predict 3D structures. So we'll talk again about this later, but here you have a page where you have all these metrics and what they represent essentially, and you have some slight or big variations between them, so they bring different types of information. So every measure has its pros and cons. So how can we establish the correspondence? We have structural alignment algorithms which are used to establish this correspondence, and this is prior to computing metrics such as the RMSD, for example. So let's say we have protein A, protein B. A naive strategy would be, well, it's not so naive, but a strategy, let's say, which is essentially what's implemented in Mammoth, will be first to identify some segments which are of similar shapes, okay, between the two. Then try to combine these segment pairs and extend the alignment. So use these segment pairs as seeds, and then extend the alignments in 3D. So how do we do this? Here you have an example where the pairs, the segments are defined by 4 atoms, okay? Can be N peptides, secondary structure elements. So here it's 4 atoms, so 4 residues. This is 4 residues, not atoms, otherwise it doesn't make much sense. So you see a schematic representation of these segments A and B. And they're just the distance between them is computed by projecting them on a circle. So you see we pick up A2, for example, B3. We're going to compare these 2 residues, and for this we just look at the edges that go from A2, the edges that go from B3, the geometrical orientation of these edges, and we project them onto the circle, and we see what we have to do to go from one green to one red, 2 green to 2 red, okay? And then to combine, you have a combination problem. So you want to find the optimal alignment. It's a combinatorics problem, but you have to sometimes make some sacrifices on the pairwise alignments. That's just like sequence alignments. So here you have an example where you have a matrix like sequence alignment matrix, and you will use this URMS metric to find the best path, which is the optimal alignment. Okay, and then you can extend by refinements. Another one is the Daly alignment. So here what you do is you compute distance matrices that correspond to each structure, which correspond to intra distances, and you try to align these matrices to put them in front of each other. So here you rely really on the description of the protein within itself to align them. And the similarity measure is here. So I will let you go a bit through these slides, trying to find also by yourself what is the DALI about, what is Mammoth about, and we can then talk about this and interact if you have questions. So here it's a good example where you have these 1 and 2, where the connectivity is different. So you see you have the same secondary elements, but in the matrix of contact, it's completely different. You have something at the top left on one case, and the squares are all over the place. So this means that you have correspondence, but not at the same place in the sequence. So you need to cross basically the sequences to make the correspondence. So this also shows that sometimes with structural alignment, you can do stuff that you will never find with sequence alignments. So here is a list, non-exhaustive because there has been more development since then of structural alignment tool. An optimal solution, you want to minimize the RMSD obviously, but then it can be different. So this is the Voronoi method. So here what is used is Voronoi diagram and Delaunay tessellations. And so these type of representations are used for many things in protein structures, and in that case it works relatively well. So each residue cell contains by definition all points that are closer to the corresponding CB atom than all the other input nodes. And so you have this definition of contacts, and then you can just take each cell and look at its environment and compare the different environments, the neighbor sets. Okay, so here are the scores. There is also some secondary structure score to improve the alignment. You tend to favor secondary structures that are the same to be aligned. So here I give you some reference which is quite recent and which compares sequence alignment and structural alignments. And what they show is that structure-based alignment is generally more accurate than sequence-based alignment, and then hybrid methods. So that's interesting. And the two best performing programs are Mammoth and Matras. Okay, so with this, I thank you for your attention, and we can talk more face-to-face about this course.